myself Dr. Deepika Seni from Lloyd Institute of Management and Technology. Today we are going to complete this parasympathomimetic agent, the last part of it of unit 3 medicinal chemistry, course code BP402T. So, in the last class we have studied about the classification, and in the classification we have studied the direct acting cholinergic drugs. So, when we talk about the direct acting cholinergic drugs, means those drugs which has acted upon the receptor directly. They were binding directly on the receptor, either the nicotinic or the muscarinic receptor, and will possess its activity same as that of acetylcholine. So, now when we talk about indirect acting means they are not directly binding on the receptor. So, they are indirectly binding. Indirectly acting means they are cholinesterase inhibitors. We can call them cholinesterase inhibitor. So, what happened when we talk about acetylcholine? Acetylcholine is being hydrolyzed by this cholinesterase enzyme. And after the hydrolysis of this, acetate and choline part will get separated, and hence the activity of acetylcholine is abolished. So, our target to develop this indirect acting cholinergic drug is to inhibit this cholinesterase enzyme. So, these indirect acting cholinergic drug will inhibit the action of cholinesterase enzyme. Hence, it will not be able to hydrolyze acetylcholine, and hence there is increase in the concentration of acetylcholine, and that will possess its action. So, these drugs actually do not act on the receptor directly, but inhibit the hydrolysis of acetylcholine by the acetylcholinesterase enzyme, and hence increase the concentration of acetylcholine so that it can produce its action in the body. Again we have studied about it in a very short, like these are of two type, one is reversible, another one is irreversible. So, indirect acting cholinergic drugs will inhibit the action of cholinesterase enzyme and will increase the concentration of acetylcholine so that acetylcholine can possess its action. So, first of all, the reversible inhibitors. As the name suggested, these agents will bind reversibly to the acetylcholinesterase enzyme, and hence they will form esters like carbonate, phosphate, and will remain with the enzyme. Okay. So, these enzymes will, what will happen when these agents they has already bound to the cholinesterase enzyme. Now, this cholinesterase enzyme is not able to exert its own action. And what was the action of a cholinesterase enzyme? To hydrolyze the acetylcholine. Now, this enzyme is not able to hydrolyze the acetylcholine, and hence these drugs we can say they can act as anticholinesterase. Anticholinesterase, sometimes students get confused in anticholinergic and anticholinesterase. Anticholinesterase drugs are cholinergic in nature, fine. Anticholinergic are which are acting against acetylcholine, but anticholinesterase they are acting in sync with the acetylcholine, because they are not acting on the muscarinic or nicotinic receptor, but they are enhancing the probability of acetylcholine to get bind with the receptor and to exert their action. So, these drugs, these reversible inhibitors, they are known as both reversible and irreversible inhibitor. They are known as anticholinesterases. Anticholinesterases are actually cholinergic in nature. So, we can say like anticholinesterase are those drugs which act on cholinesterase enzyme to inhibit its action of hydrolyzing acetyl coenzyme acetylcholine, and will hence increase the concentration of acetylcholine, and thus also produces like it will if it has increased the concentration of acetylcholine, means the acetylcholine is free to produce its action. So, the first example is physostigmine. This is the structure of physostigmine. It will indirectly stimulate both muscarinic and nicotinic receptor, because it has indirectly. Why we are saying like again indirectly stimulate? Because it has inhibit the action of acetylcholinesterase. So, now acetylcholine is present there. So, acetylcholine can act through both the receptors, to the muscarinic receptor as well as to the nicotinic receptors. It is used to decrease the intraocular pressure in the glaucoma. But the point is these drugs will produce the similar action. The direct acting drugs, they were also used to decrease the intraocular pressure, as we have discussed in the glaucoma. There is a glaucoma is a diseased state where there is increased intraocular pressure. So, the point is we need to decrease the intraocular pressure. So, with the help of physostigmine we can do that. That will bind to the, though it will bind to the cholinesterase enzyme, but the release of the concentration, high increase of acetylcholine, that will definitely help to reduce the intraocular pressure. Next is neostigmine. This is neostigmine. The structure is this. So, the IUPAC name is 3-dimethylamino carbonyl. This is dimethylamino carbonyl oxy, this is oxy, NNN, means there is 3 substitution at the N and that is similar, that is trimethyl. So, here we can see NNN, N-methyl, N-methyl, N-methyl. So, NNN trimethyl benzene, this is benzene, ammonium, this was the ammonium, and the salt is bromide. So, the overall the name is 3-dimethylamino carbonyl oxy, again 3-dimethylamino carbonyl dimethylamino carbonyl oxy NNN trimethyl benzene amino ammonium and bromide. Fine. So, this was the IUPAC nomenclature for the neostigmine. It indirectly stimulate both, again the same point, because it will indirectly stimulate both nicotinic as well as muscarinic receptor. As we have already discussed, it will what will happen? They will bind to the acetylcholinesterase enzyme and increase the concentration of acetylcholine. So, indirectly they are stimulating muscarinic as well as nicotinic receptor. Now, they will bind to the anionic and ester site of cholinesterase enzyme, and hence what will happen? They are binding themselves to the, either they will find anionic and the ester site, they will bind to that. Now, the cholinesterase enzyme will not be able to possess its action. So, cholinesterase would not be able to hydrolyze acetylcholine. and here is the synthesis of neostigmine. So, first of all what we will take metachloro-N,N-dimethylamminobenzene. So, first of all when we talk about this is benzene, its amino, it means it contains N, and at N it is N dimethyl. So, it has dimethyl, this is dimethyl again, and metachloro. So, this is first position, these are ortho, these are meta. So, either side, either of the side कहीं पे भी लगाएंगे, this is chloro. So, metachloro NN dimethyl. So, metachloro NN dimethyl amino, and this is benzene. So, by the action of KOH, what will happen? This potassium will replace this OK, oxy potassium will replace this chlorine atom, and hence there is a potassium salt of it. Further, dimethyl amino carbonyl chloride is taken as one of another reactant. Now, we have dimethyl, this is dimethyl amino, this is carbonyl, and this is chloride. What will happen? This KCl will get removed, and this the whole group will be incorporated with along with the oxygen. So, OCONCH3 twice, it is as it is incorporated. Further, by the action of the methyl bromide, now this amine group, this has been converted into ammonium salt, the quaternary salt, and there is a bromide salt of neostigmine is present, is formed. So, this is neostigmine bromide. So, what has happened? First of all, let us start with the metachloro-N,N dimethyl aminobenzene. By the action of KOH, we have replaced this chlorine with the OK, this is oxygen, and because the KOH here, this chlorine is being replaced by OK. Further, this dimethyl amino carbonyl chloride, and there is a removal of KCl, and there is this group will be attached OCONCH3 twice. Further, the methyl bromide is added, and that will lead to the formation of neostigmine bromide. So, there are another few examples of reversible anticholinesterases like pyridostigmine bromide and edrophonium chloride and ambanionium chloride. So, when we talk about the pyridostigmine bromide, it is also helpful in the nerve gas poisoning. What happened before surgery, as we have discussed, we have taken anticholinergic drugs, and sometimes what happened, there is poisoning of those drugs, means there is toxic effect of these drugs. In that cases, we have to use reversible anticholinesterase, like the example is pyridostigmine bromide. Further, this edrophonium chloride, this is also used in the treatment of snake bite. In the case of snake bite, ophthalmoplasia will happen, that is a very painful condition for the eye. So, in that case, this drug really helps, and all these drugs are used in the treatment of myasthenia gravis. What happened in the case of myasthenia gravis? Skeleton muscles are completely relaxed. Completely relaxed means the patient is not even to move through those skeleton muscles. So, what will happen by the acting through these reversible anticholinesterase drugs? So, what will happen, there is increase in this muscle, because these muscles has been relaxed and they are there is no movement, we can say. So, to improve the movement, what will happen, we need to contract these muscles, and that will happen through the nicotinic receptors. So, these all drugs, all the reversible anticholinesterase drugs, they also used in the treatment of the myasthenia gravis. Further, irreversible cholinesterase inhibitors, the next category. So, these are the drugs that will produce, that will inactivate the acetylcholinesterase enzyme, but their binding is irreversible. Irreversible, they will bind to the enzyme through the covalent bond. So, that is the reason they are irreversible in nature. So, bind irreversibly by the covalent bond to the active site of the enzyme and hence lead to the inactivation of these enzymes. Further, this category include different type of organophosphorus compounds that we have already discussed in the previous lecture, as well as when we have seen the structure of these irreversible cholinesterase enzyme, we have noticed there was these are the organophosphorus compounds. So, let us take some examples like malathion, it is used in the insecticide, again organophosphorus as we have discussed, and also used in the treatment of the scabies. And further, these are also parathion, one of the most common insecticide used in agriculture, and isoflurfate and ethothiopate iodide. These are the examples of the irreversible anticholinergic drugs. So, the next topic is cholinesterase reactivator. So, sometimes actually what happens, we have used indirectly acting cholinesterase inhibitors, like we have say. So, indirectly acting acetylcholine or cholinergic drugs, what they did, they had bind to the they have a tendency to bind with the cholinesterase enzyme and they inhibit the action of cholinesterase enzyme. Sometimes this cholinesterase enzyme is to be reactivated. Why reactivated? Means there is poisoning of those drugs, poisoning, or we can say there is excessive amount of those organophosphates or the sulfonates or the drugs that those inhibitors, there is a poisoning of those inhibitors. So, in that case what do we need? We have to reactivate our cholinesterase enzyme. So, when we talk about cholinesterase reactivator, these are the drugs which will cause reactivation. Reactivation means now the cholinesterase enzyme will be reactivated to produce its hydrolysis action. So, to produce that hydrolytic action, we need this cholinesterase enzyme, but this cholinesterase enzyme is bound with the irreversible or the reversible, we have studied earlier, the inhibitor, the acetylcholinesterase inhibitors. So, these reactivators will help to treat the poisoning of these inhibitors. Means after a prolonged use of this acetylcholinesterase inhibitors, now the activity of cholinesterase enzyme is being abolished for a period of time. Now that is producing a problem. So, to treat that poisoning that is being produced by the organophosphates, sulfonates or acetylcholinesterase inhibitors. So, now what will we do? We will use cholinesterase reactivator, and the example is pralidoxin chloride. The pralidoxin chloride is a cholinesterase reactivator, and the name is 2,4-1-methyl-pyridinium chloride oxime. So, this is the IUPAC name of pralidoxin chloride. It reactivates the enzyme by binding to the anionic enzyme and displays the phosphate from the serine residue. So, what has happened, as we have studied earlier, in case of inhibitor, cholinesterase inhibitors, that they will bind to the anionic and ester site, so that they can stop the activity of cholinesterase enzyme. So, now what will happen? Pralidoxim will also bind to the anionic site of the enzyme and will remove the action of the organophosphorus compound that has inhibited the action of cholinesterase enzyme. So, it will reactivate the pralidoxim, it reactivates the enzyme by binding to the anionic site of the enzyme and displaces the phosphate from, so that the phosphate is being displaced from the serine residue. And the main function of this pralidoxim is to treat the poisoning of the organophosphorus compounds. The organophosphorus compounds, those which has produced the cholinesterase inhibitor action. So, here is a quick revision of these drugs, like indirect acting drugs. These indirect acting drugs are the drugs which will not directly bind to the either to the muscarinic or to the nicotinic receptor. So, what we need to do? We need to increase the acetylcholine concentration. What will we do? We will inhibit the action of cholinesterase enzyme, that is the cholinesterase enzyme which is known for its hydrolytic property of acetylcholine. So, after the inhibition of hydrolysis of acetylcholine, so hence that will increase the concentration of acetylcholine and it will produce its action. Two types are there, reversible and irreversible. So, when we talk about reversible, these agents will bind reversibly. So, once they will bind to the enzyme and after possessing its action, they will leave the enzyme. So, they are anticholinesterases and the example is physostigmine. They are used in the treatment of the glaucoma to reduce the intraocular pressure, and the neostigmine is also bind to the anionic and ester site of the cholinesterase and block the activity of acetylcholinesterase. And in the case of synthesis, we have started with the metachloro-NN dimethylaminobenzene, led to the further with the action of the potassium hydroxide and dimethylaminocarbonyl chloride, we have synthesized neostigmine bromide. Further, there are some the examples of some reversible anticholinesterase. Here we have studied, like all of the reversible anticholinesterase are known to for the treatment of the myasthenia graviasis. And further, irreversible cholinesterase inhibitors, these and irreversible cholinesterase inhibitors, they bind irreversibly because they bind through the covalent bond and they will cause the inactivation of the enzyme. And in this category, organophosphorus compounds are there, like malathion, parathion. Basically, they are used as the insecticides and also in the treatment of scabies. And there are some few other examples as well. Further, sometimes we need cholinesterase reactivators because of the poisoning of the organophosphates that we have studied earlier, as they are binding to the enzyme irreversibly, making it inactive. So, sometimes we need to reactivate the cholinesterase enzyme to reduce their effect. So, there are the example is pralidoxim chloride. It will reactivate the enzyme, it will again bind to the anionic site of the enzyme and will replace the phosphate from the serine residue. So, basically they are used in the treatment of the poisoning of the organophosphorus compound. Thank you very much.